This is my institute in La Jolla. It's on the UCSD campus. It's the first carbon neutral research building in the world. You can see the roof is solar panels. It's a 0.5 megawatt power plant, and 5 years of unique engineering went into this building that has 2 gigantic water tanks, that one for hot water and one for cold water. Engineering principles show that it's a lot more efficient to move water than to move air. So instead of giant air ducts, we pump water around with heat exchangers in each room. The windows actually open, but they're computer controlled, and there's computer controlled blinds in every office, and every lab has windows. So it's a very unique environment, and people seem to really like it there. So I've been pursuing this question, what is life? And I brought a unique lens to that question, the lens of genomics. And that led to multiple other questions. Can the genetic code actually predict present and future phenotypes? Ultimately, can we rewrite that code? And even basic questions, does DNA contain all the necessary information for life? This was all hotly debated. And what is its diversity? So I started at NIH randomly sequencing cDNAs. I named these ESTs. And this work was actually inspired by a trip I took to Japan. I was asked to, as a member of NIH, to go with the State Department to review technology in Japan that was being developed to sequence the human genome. There were multiple claims, and the Japanese government was giving industry very large grants to make machines and robots. Some of these were quite comical. Seiko robot arm, if anybody's ever seen it working, they use them to build robot watches. Instead of developing something new, they copied what scientists did. This beautiful, expensive robot would pick up a toothpick, stab it in a colony, and put it in a tube. So, the design of robotics is doing something not following what humans do. So those things never progressed. But I had great discussions, and I became good friends with Kenichi Matsubara, who became head of the Japanese genome effort. And he said he was concentrating on cDNAs. And that's where I got the idea to just randomly select them and sequence them, because I had the first automated sequencers. But as we read the genetic code, I call that digitizing biology, because we're converting the 4 base code into ones and zeros. Aside from ESTs, which we did by the hundreds of thousands, and what was ultimately used to annotate the human genome, Smith and I decided to apply this to a genome. It involved developing new algorithms to assemble DNA, to sequence Haemophilus. By the way, Hem and I wrote a grant, submitted it to NIH, and it was turned down with extreme prejudice. This idea could never work of assembling things. We had to sequence 25,000 sequences, roughly 500 letters long, and the algorithm was able to stitch those together and reassemble the genome. I was certain that this would be scalable, and 5 years later, we scaled it up to sequence 25 million sequences and assemble those with even an enhanced algorithm for the human genome. And the announcement of the White House was totally based on when our computer run finished, and we said we had a genome. They would call me daily, getting very nervous, because they schedule these events in advance, and they're worried the genome wouldn't be ready in time. The public effort, which spent $5 billion versus our $100 million, didn't have anything until the following year, but we called it a tie to be politically correct. In 2000, the early genomes were just haploid genomes, and almost everything that's been sequenced is a haploid genome. you would not be alive if you only had a haploid genome. And so we wanted to see how much more complex a diploid genome was. And so Sam Levy at the Venture Institute at the time, so my institutes, Tiger and others, all merged into one, expanded from the Celera data. This is probably the only genome that's been sequenced with Sanger sequencing because the transition to new techniques was happening at the same time. But this showed that we had far more variance in rearrangements, insertions, deletions than just SNPs. So we started with Haemophilus. After sequencing Haemophilus, even though we couldn't get funding for it, then we were offered tons of money to sequence every pathogen, every genome on the planet. The Department of Energy set up a special committee to advise us which ones we wanted to do. We got a clinical sample from Norway from a patient not responsive to therapy. And on sequencing the sample, the algorithm assembled it into two separate species. And that's when, on top of the 25 million sequences from human and doing the Drosophila genome, the mouse genome, I knew that the algorithm was so powerful and that it became clear there was a unique mathematical solution for each species genome, and in fact, for each human genome. And so I got the notion of sampling much more complex environments. Again, we wrote a grant, submitted to the Department of Energy, and the reviewers shredded it. But Ari Petrinos, who was head of biology at Dewey, had the ability to override reviewers. He was a believer in large-scale computing with large-scale biology and wanted to fund it. And fortunately, it worked out. We filtered a barrel of seawater off of Bermuda. We have these 3 different filters to collect different size organisms from viruses up, and then the filters could just go in a freezer and then get shipped back to the lab, all the DNA isolated and sequenced. And so just from the first barrel of seawater, when there was not supposed to be any more than a dozen or so organisms, we discovered around 1,800 new species. We stopped sequencing about 1.5 million new genes, and it just opened everybody's eyes, and the world overnight was very different. In the Sargasso Sea, and those of you who ever did molecular biology, before genomics, the tools of molecular biology were a microscope and agar media. If it wouldn't grow in culture, it didn't exist. And so organisms were numbered one at a time as they were discovered in the ocean. They were up to SAR 11, Sargasso Sea 11. And somebody published a paper showing how little diversity there was in the oceans. And as a sailor who was, after sequencing the human genome, I took off 6 months and sailed around the Caribbean and recovered my brain from all the press and trauma of getting the genome sequenced in 9 months, and was reading these papers about the lack of diversity, and decided to do, that was the impetus for doing this experiment. So the question was then, do we just keep sampling this one site, or was it likely that another site would be different? Was the ocean a giant homogeneous soup? You know, being a sailor, people think my best idea was combining sailing around the world in my own boat and doing science. I think it was a very good idea. It was an incredible experience. We largely followed the Challenger expedition, which started in Halifax, stopped every 200 miles, sent a dredge down to the bottom to see what the bottom of the ocean was like. So this was the first scientific expedition. It was in 1870s. And the brilliant scientists at the time said there couldn't possibly be any life below 1,800 feet. But every place Challenger put down their drogues, they found life in the deepest trenches even, and changed that view. So it was very successful, but they, like Darwin, could only see macro life. We covered most of the major seas, went up into the Baltic, the Mediterranean, the Aegean and Black Sea. One of the biggest discoveries was that instead of individual discrete species, we found these clouds of organisms. So SAR11 is a photosynthetic bacteria found in the Sargasso Sea. So these plots are by taking the reference genome, sequencing the SAR11 genome, and then from the millions of fragments that we had, seeing what aligned with that genome. And that's what shows on these plots. And you can see they're actually huge clouds. This area here is where the reference genome had a viral insertion that wasn't shown up in most of the other organisms. But these varied by as much as 50% sequence identity. All mammalian species differ by less than 10%. So this is tremendous diversity, but gene order was largely maintained. And the reasons for this is there's two major drivers of evolution and mutation, UV light and oxygen. These organisms produce 50% of the oxygen we breathe, and they're constantly exposed to high levels of UV light. So think of, and we found major clouds in that environment. We published the first analysis on describing over 6 million new species. And again, every time there's a bottleneck in thinking that we think as a community that we know everything, that's sort of when major discovery really starts to happen. We thought we knew all protein families and protein folds. Just from this data set, we discovered thousands of new protein families and multiple new protein folds. So this was a special issue of PLOS biology. We also sequenced a lot of viruses. So there's 10 viruses for every microbial cell. They help control the population in the oceans of their host. But we discovered something even more important. They do a lot of lateral gene transfer, and viruses carry new versions of photosynthetic enzymes, and they constantly upgrade the software in their host. They want their host to be alive and robust. Viruses don't want to kill off their host. And so it kind of raises a philosophical question if viruses are trying to keep their host alive to reproduce, who's really at the top of the food chain, and how is the system evolved. We also looked in the deep sea from high temperature vents. That's where this organism, Methanococcus unaceae, was. This was the first archaea genome that we sequenced. So this lives temperature optimum is 85 degrees centigrade, over 200 atmospheres of pressure. And its genes were sort of half prokaryote and half eukaryote. So this was collaboration with Carl Woese and clearly proved his hypothesis that archaea was a third branch of life. It's interesting with deep organisms, and this is sampling a mile deep in some coal bed methane mines in Colorado. We see a lot of species diversity, but this is the same kind of plots I showed you from SAR11. You don't see the clouds of organisms. These are very high temperature environments. There's no oxygen. So there's really nothing to drive mutagenesis. The closest relatives to the things Mile Deep in Colorado were from a volcano in Italy. So in the subsurface, we see lots of conservation. We don't see a lot of mutagenesis and changes. And some have argued that there's more life beneath the surface of the earth than there is on the surface. All this is documented in my book that came out last month, and I understand some will be available later. It talks about the science and the diversity in the oceans, and that there's more. We discovered more species than there are stars and planets in the universe. So it's been easy to recruit physicists and planetary science now to biology, because biology is far more interesting, but also has adventure stories of we got arrested numerous times and all the problems with sampling around the world. And we got caught in geopolitics of various countries fighting over things that had nothing to do with science. So I think you'll find it to be an enjoyable science and sailing adventure read. One of the fundamental questions we're always trying to answer, does DNA contain all the necessary information for cellular life? Comparative genomics, we thought, would help give us that answer. We decided to take Richard Feynman's approach and to try and build it. And to do this, we go the other way. We take the digital code out of the computer, 4 bottles of chemicals, and rewrite the genetic code. This started slowly in 2003. We decided to start historically with Phi-X174, which is the first virus that Fred Sanger sequenced. Clyde Hutchinson, who was an emeritus professor at the Venture Institute at the time, was actually in Sanger's lab at the time and always carried a vial of Phi-X174 in his pocket, which always seemed a little strange, but we had a reference strain to look at. We tried just making the overlapping oligonucleotides and seeing if we could PCR them up together. Even with selection by infectivity, we didn't get a single viable virus doing that. And so the machines that synthesize DNA, the oligonucleotide synthesizers, are fairly degenerate. The longer you get, the more errors. So we had to develop methods of error correction to correct the errors made from the synthesis. When we did that, this was the first experiment where this is a lawn of E. coli, just adding our synthetic DNA. DNA is infective with Phi-X. So when you see these clear spots, that's where the virus replicated and killed other cells. So we call this a case where the software builds its own hardware. We just put in the DNA software and it took over cellular machinery and started making physical copies of itself. But we actually wanted to make a living self-replicating cell. So that started the 10-year process to make the first synthetic cell. We had to develop all kinds of techniques. I won't walk you through them, but it was a long, arduous road to get to making megabase size molecules. And the largest piece that had ever been made was 30 kb. So getting up to 1 million base pairs took a lot of new techniques. DNA at this size is so brittle, you can't pipette it. And we have to move it around electrophoretically in and out of gels. We can process it while it's in gels, but it becomes important a step to learn how to get them to the next stage. So we had 2 teams, one working on the synthesis and one working on transplantation. So I had to get to transplant sooner or later here. So John Glass and Carol Latigue are the two leaders of being able to transplant genomes. And by transplanting a genome, we're able to convert one species into another. This was a huge step, and we never would have gotten to a synthetic cell without this process working. It's still a fairly specialized system, but in the design of the genomes, we can control which restriction enzymes are there. Ham Smith, if you recall, got the Nobel Prize in 1978 for discovering restriction enzymes. So we had somebody in the team who knew something about them. So we insert the new synthetic genome into the recipient cell, and it starts expressing genes. We think the restriction enzymes then recognize the parent chromosome in the cell and chew it up. So now we have a body of one cell and the DNA species of another, but it rapidly converts all the characteristics of the cell. There's not a single molecule of the parent cell left. It's an entirely new cell driven by the software that we inserted in it. So this was 2010. It was a big global announcement. People thought this was going to cause religious riots and all kinds of problems. We had a press conference in Washington, D.C. Every federal agency was there. They had the building surrounded by police. They were just totally afraid of this announcement, and it went much better than they expected. I got comments from the Pope and the President on the same day. President Obama formed his new bioethics commission to review making synthetic life. And the Pope commented, he said, Craig Venter did not create life, he just changed one of life's engines. I couldn't find life's engines anywhere in the Bible, but I'm glad we could change one. Now, to make synthetic life, we didn't want it to be confused with anything else, so we designed a totally new code to write the English language with punctuation and numbers in the genetic code. Now, people have used ASCII code to do this, so it wasn't an original idea. But we designed a code that puts in very frequent stop codons because we wouldn't want to put in University of Toronto and have that turn into a new biotoxin. So we're able to write all these things, but none of these would be translated into proteins. We listed the names of all the scientists, institutions, and then I added 4 quotes from the literature. I don't know if you can read those. The first from James Joyce: to live, to err, to fall, to triumph, to recreate life out of life. The second is from Oppenheimer's biography from an early teacher: see things not as they are, but as they might be. And the third, more important, is Richard Feynman: what I cannot build, I cannot not understand. We also put in a URL, and it was a contest to whoever could decode this first and send an email to the bacteria won a prize. So there was all this built into the genetic code. We're quite surprised. Some of the first comments we got were contacted by James Joyce's state attorneys asking if we had permission to use the quotation. Under US copyright law, you can use up to a paragraph as long as it's proper attribution. And then I started getting emails from scientists at Caltech saying I had misquoted Richard Feynman. And if you go on the internet today, this is the quote you find. And so finally they dug into the archives, sent me a picture of Feynman's blackboard. And so the original quote was, what I cannot create, I do not understand, which works equally well in our case. And the whole reason to write the genetic code was to understand it. Now, for the first version, and we call that JCVI 1.0, it was largely similar to existing cell mycoplasma mycoides. We made some differences, but our goal was to actually design a minimal cell from scratch and see if we could design and build it. And so we had a contest where several of us came up with our genome design. We made those chromosomes, not one of them worked. And so we had to start adding back genes to find out what was missing. And so we had this design build process where we design a chromosome, test it, and go through this cycle. And so we had 3 major cycles, and the problem was there were a number of unknown genes that were absolutely essential for life that had to be in the genome to get a living cell. So we finally, after numerous cycles, got there. But this genome, it's the smallest genome of any species in history, has 473 genes, but 149 of them are of unknown function. So it's hard to design things de novo based on first principles when we don't know the first principles of roughly a third or a quarter of life. And it's also part of the intellectual bottleneck, of, again, we thought we knew everything, and we showed now in every species major genes of unknown function. So this is the minimal cell, JCBI SYN 3.0. It's made billions of copies of itself, but in trying to understand the genome and cellular life from looking at the genetic code, we have this problem of unassigned, unknown genes that provide essential pieces of life. Because we use computer terminology, we also tried to defrag the genome, organizing it. You can see it took a lot of changes to do this. But the amazing thing is we could reorder things, put them in a logical order, and things still functioned. So the random order of genes is not essential, but what is essential is to have the right regulatory partners with the genes. But this should make the process of design easier in the future. So the minimal cell has sort of become the research tool for evolutionary biology. There's 60 major labs around the world studying it, and more papers are coming out on it since our first publication. Our biggest goal initially was to understand the 149 unknown genes and what their functions were. And you can see they're highly conserved throughout certainly the bacterial kingdom. But we just have this whole block using numerous techniques, including the latest AI protein predictions, have helped get it down roughly half. We're down to 78 genes of unknown function. And some of these are pretty amazing. So the phenotype of these cells in the most minimal form were really unusual. The normal wild type cells are on the left, but we'd get these giant cells as well. And so this is the SYN.1, the wild type growing. It divides normally, creates lots of individual daughter cells. Everything looks normal with DNA, membranes, and cytoplasm. But SYN 3.0 lacks cell division machinery, apparently. And we get these just giant blobs. And we think we've basically created a primordial cell that clearly has DNA and membrane. When we added back the known genes for cell division, that did not work. In fact, we ended up having, by trial and error, adding back a set of 7 genes, including ones of unknown function, to restore normal cell division. So every cell on this planet divides by the mechanism shown on the left. The SYN 3.0 without the 7 genes sort of divides like Jack Sozak showed with, he was trying to make primordial cells with lipid membranes, and they sort of separate like bubbles. You've all made bubbles and you see how they turn into things. We think this is, we got back to how early cells divided. But now the notion with this minimal cell is by adding genes back, we should be able to recapitulate all of evolution. So adding back those 7 genes restores normal cell division. People have been doing computer modeling of the cell, and this is what we think it looks like inside. It's not a bag of jelly. There's lots of copies of the chromosomes, lots of copies of the ribosomes. People don't realize that each of us is alive because your genetic code is being read every second. Proteins have a half-life of seconds to minutes, very few last hours. So you take DNA out of a cell, cells die very, very rapidly, with the exception of red blood cells. That's why radiation damage, knocking out the chromosome, kills people. If you block this process of going from the software of DNA to making proteins, cells disappear very rapidly. So I argue that life is a DNA software system, and we've shown simply by changing the software, you change the species. So we were transplanting DNA, not all organs, but we wanted to get back to understanding the human genome. You know, the diploid genome, non-SNP variants count for 75% of the bases that differ between individual genomes. This was my genome. My 2 parental chromosomes were completely phased by sequencing single sperm cells, sperm being haploid. We were able to completely phase the chromosomes. And there was a 0.5% difference between my 2 parental chromosomes. So if you think about the complexity of this, for every gene in your genome, you have potential for at least 2 different copies, producing 2 different proteins. And so the diversity goes up quite tremendously, but these get regulated quite independently. For example, a simple test, I don't know how many of you get jittery if you drink a cup of coffee or tea. If you're a surgeon, that's not a good thing. So there's 2 genes controlling caffeine metabolism. There's 2 slow versions and there's 2 fast versions, or you can have interchange copies. I have 2 copies, and like I might have an extra copy of the fast one. So I can have 2 large mugs of strong coffee in the morning, wash it down with a Red Bull, and not be the least bit jittery. The other extreme, that's enough to cause lethal arrhythmias in people that are slow metabolizers. But everybody sort of knows who they are, whether, you know, coffee or tea or caffeine makes you jittery or not. And surgeons learn this early on and learn not to drink coffee or tea before surgery. But we were trying to understand this, and once we sequenced 10,000 genomes at human longevity, and we're able to look at over 1 billion data points to try and understand the genome. And there's areas that are very susceptible to mutation. For example, where you see the arrows, if you have a mutation at any of those points, it's incompatible with life. So just random mutations on those points could be what causes spontaneous abortions, early death. You can see there's areas that are highly sensitive to mutations and others that are not. Some of these are very logical. If you look at all the transmembrane proteins, and we looked at almost 24,000, the transmembrane point, the point that sits in the membrane, is highly susceptible to mutation. This makes sense. If you change from a non-charged to a charged amino acid, it's going to throw that protein out of the membrane and knock out its function. So we're getting areas and can scan the genome and know areas that are susceptible to mutation and ones that are not. Now, here's one of my pet peeve slides. The FDA thinks mice are little humans. This is looking at essential genes in mice versus essential genes in human. And also the other mistake we make is thinking that haploid cells in tissue culture are mini versions of humans. You can see with this Venn diagram, there's very little that carries over from life in culture to mice to essential genes in humans. And the essential gene population is important because when we're looking for pathogenic variants, they show up much more readily in essential genes. You guys know this data probably better than most. These are the most likely reasons you're all going to die. Hopefully not today. The numbers for females are changing with greatly increased numbers lately in cardiac deaths for females. We have an interesting national view in the US of this. So this is the map of male longevity. You can see the South is much lower, so it's not healthy to live in the South. But we have these other red patches. Those are primarily indigenous population tribal lands where the healthcare and the longevity is substantially lower than the rest of the country. To show you that genetics is important, this is what happens when you have two X chromosomes. It's a much healthier picture of the world, still the South and those same patches, but it looks much healthier picture for women. So accidental deaths are constantly going up, but this is primarily due to all the accidental poisonings and drug overdoses. Unfortunately for transplantation, car accidents are way down, which used to be the major source of transplanted organs. All the ones dying from fentanyl and other things are probably not good. Falls keep increasing. And this just shows the curves, the populations drop off and not a lot reach beyond a century. We think these curves can be moved to the right with pre-symptomatic testing. I have the secret to life. It's not to die prematurely from accidental death or preventable early detectable disease. Now, I have a different version of this than most physicians. And we've shown that diseases can be predicted and detected before they become clinically apparent. We started by measuring comprehensive human phenotypes in doing this in non-invasive, except for one blood draw. One of the biggest advances is in post-processing of MRI data using restriction spectrum imaging. So tumor cells have larger nuclei than normal cells. They actually have more water, but the water is less mobile. So it sends a very strong MRI signal, as you see in that bright orange at the bottom. This is a study on prostate cancer using normal MRI and then the restriction spectrum. You can see that the tumor areas just totally light up with this new post-processing algorithm that corresponds nicely with pathology. We set up at Human Longevity just to see people with no symptoms, people who thought they were healthy. I was the first one to test the system, and as we were building the clinic, I was told by the best urologist in San Diego that I did not have prostate cancer. My pathologist friends came and told me after testing the first MRI that in fact I had 2 high-grade prostate tumors, and that if they weren't found by this happenstance testing, I would have been dead within 5 years. So Leonardo da Vinci did my surgery. He did a great job, and I've been cancer-free for almost 7 years now. The second was I paid for all the employees of the Venture Institute to go through the health nucleus. This was Ham Smith's lungs. He had a very rapidly growing lymphoma. He had no symptoms. Surgeon said he would have been dead within 6 weeks if we hadn't found this. He just celebrated his 93rd birthday. He does have some residual effects. They couldn't surgically remove much of the tumor because it was around the esophagus. They used radiation therapy, and he has some damage to his esophagus. But the 2 guys that sequenced the human genome are both alive and well because we decided to do this testing. We find high-grade cancer rate of roughly 5% of everybody 50 and older with no symptoms. So you guys can do the math even with this size audience. One of the nice things with the MRI, it gives us organ fat, particularly liver fat, for diagnosing metabolic disease. I was on a halfway through a 30-mile bike ride with the world's expert on organ fat. And then we stopped after 15 miles and we were just talking about it. He goes, you know, this exercise won't lower your liver fat. That you have to, only calorie restriction is the only thing that will change your organ fat levels. And I said, why in the hell didn't you tell me that 15 miles ago? But this is one of the most used diagnostics coming out of this. And I went from very elevated liver fat. I've lost 65 pounds, and now I'm down in the low end. But obviously a lot of NASH is detected. So just from the MRI, we're able to detect fatty organ disease. We find this in 23% across all age groups of asymptomatic individuals. So roughly a quarter of you have a fatty liver right now, which is not good for your longevity. We get this beautiful non-contrast angiography looking at brain blood vessels, and we find things like this. So aneurysms, some of them are quite large, and we find these in 1% of the population, but the majority are under 50. So what does that mean? I mean, genetic selection has killed off people over in that they died from their aneurysms. What's the major symptom? You report to the ER with a massive brain bleed. Usually they're lethal. These can now be treated as an outpatient in roughly 15 minutes. So this is totally a preventable death. We get comprehensive data on the brain with the new brain volumetric analyses. So we get hippocampal volume of different areas. And this associates pretty clearly with Alzheimer's risk. But the original motivation from this was my genome, which I'm heterozygote for ApoE4. I asked to get a brain MRI, and they wouldn't do it because I didn't have any symptoms. But they agreed finally to do it in a pseudonym, and they said I had the brain of a 40-year-old. So being heterozygote shows up almost nothing on the MRI. Homozygote ApoE4 show up about 82% of the time. So it does mean something. We're able to predict roughly 17% now from genetic analysis combining with the phenotype data. But the main issue needed for understanding the genome is correlating it with phenotype information. And I made some radiologists very happy. I said, if I was going to select a single test, I would choose whole body MRI over having my genome sequenced. But the combination of the two and the combination with these other things in pre-symptomatic individuals is really turning up some stunning data. We're approaching 10,000 patients, and we'll be publishing that analysis, but basically the same statistics are holding up. Roughly 50% of people that come in that think they're healthy, we find a major disease from pre-diabetics to major tumors. The tumors have almost all been phase 0, phase 1, and with a tremendous track record of having, like in my case, having the tumor removed and the problem goes away. I'm just going to close. I had, we've been talking about genome transplants. I have a few slides just on the xenotransplant work doing with Martine Rothblatt and United Therapeutics. My role in this was relatively small. She came in and asked if I could genetically engineer some pig cells. We sequenced the pig genome and found a place where it was safe to insert genes, not subject to lots of recombination. And we started making genetic changes. We ended up changing 10 genes on top of the gal gene that was working pretty well on its own. As you know, the first heart transplant was done last year. The patient lived about 2 months, which was longer than the first human heart-to-heart transplant. The second patient was just done last month. This was the patient 2 days ago. He's starting to get up and exercise. So he's a month into it. But as you guys know better than I do, it's the long-term rejection that's really the challenge. But both the patients would have died within a week or so without these transplants. The pigs are raised in special sterile conditions. We check the genome for zoonotic viruses. So that's a key part of this. One was found in the first patient. So we're hoping this one does better. The kidney transplants are going on in baboons and seem to be doing extremely well. As you know, 1 million kidneys are needed a year. That's just in the US. And so we're hopeful with trying to humanize pig tissue, changing complement and other genes that we might have more and longer successes. It's early on. Turns out almost none of this biology was known, and seeing the differences between human and pig organs, the physiology is not surprisingly different. We're trying to overcome that with continued genetic engineering. Well, with that, I covered a lot of territory, but thank you for your attention. "59 minutes, I promised.
Thank you very much. You could have taken much more. I'm sure everybody would have just sat here and listened to you. Congratulations, first of all, on all your work and on receiving this wonderful award, which we're thankful of our Japanese colleagues to provide. We have just a few minutes. Dr. Venter is actually going to be here all day. Not that you can just wander in and see him. We have scheduled visits for people. The students will have time to meet with him, but you can stop by. He'll just be over in the other room. And then tomorrow he'll be with Dr. Stephen Scheer and the group of sick kids, because he's on the advisory board of their institute. We have time for just a couple quick questions, though. Maybe I could just ask you, with regards to the liver, the NASH, the microbiome. I know you've written on the microbiome, and have you noticed changes in the microbiome, especially us in Western societies? I look around the room, I see a few people who have probably physiques which we would all envy. Most of us don't have those physiques. And so clearly with what we eat, has that influenced the genetics of the microbiome, which has then interacted with the liver in this case? Can you study that using your genetic tools?
Yeah, no, in a couple of years, we published with a group at UCSD finding interesting correlations with changes in the microbiome and NASH. You know, there's so much misinformation published on the microbiome that it's hard. It's like stem cell research. It's hard to sort out what the real facts are, what really works and what doesn't. We're taking the approach that we did with synthetic cell. We're trying to reproduce a normal microbiome. We have about 50 isolated anaerobic organisms that grow in unique culture that can be scaled up for FDA purposes, that we think can restore a microbiome. These contain most of the metabolic activity of a normal microbiome. So NASH is certainly one of the cases, but C. diff, there's 800 clinical trials going on with fecal transplants, right? And the FDA shut down several of them due to deaths. I think we can come up with something more aesthetically pleasing than a fecal transplant. And I think these pure isolated cultures from a healthy set of healthy microbiomes might provide the answer.
Dr. Kashab.
I really enjoyed your lecture. You know, one of the biggest impediments, I think, to xenotransplantation has been the fear of zoonosis and transfer of infections and unleashing infections on humankind. And I was just wondering, like, with your ability, like, to analyze a milliliter of seawater and find all these organisms and so on, will there be ways to better reassure the public about the risk of infections that might go with the xenotransplant?
So we constantly resequence this line of pigs that is grown under as clean as conditions, as you know, as possible. So we can certainly tell if there's any viral insertions in the genetic code. But as we did with humans, we can just do a whole virome analysis. And now that it's gone the first genome cost me $100 million. You can now get your genome sequenced for $200. And so the cost of sequencing and analysis, there's no excuse not to do that on every single case.
Okay.
So, trouble is most of the viral world is a giant unknown. And, but just detection of any novel viruses is key to not going ahead with that organ.
Would you see that being a quality control thing that goes with every organ that comes out?
I would like to see that, yes. And with the United Therapeutics program, they try and keep them under sterile growth conditions, and they do monitor it. And after every CRISPR edit, we resequence the entire genome looking for off-target effects. So there's probably more quality control going into what that group does than anything I've seen anywhere. But it's one thing we have to really be vigilant about. But that's why pigs are much better than non-human primates. The closer viruses are much more of a problem. But I think as we identify more and more, it'll be easy to eliminate them.
Okay, Kumar.
Yeah, thank you for that. I was really fascinated by the synthetic life piece, and I'm just wondering, it took evolution, I think 1 billion plus years to go from single cell to multicellular life. And I'm just wondering, is that on the horizon, or is that possible to go from single cell to synthetically made multicellular life?
Yeah. So, there's been other gene sets added back to the minimal cell. A group in Europe found the genes responsible for cell motility, and they added it back, and the cells started crawling around the plate. And so we have a major effort of trying to see if we can create a eukaryotic cell out of this minimal mycoplasma cell. We want to add back photosynthetic enzymes. So we're working, trying to recapitulate evolution to get to that stage. There's not a lot of gene regulation in the minimal cell. Things are basically on or off. As you get into eukaryotic biology, there's a lot more fine tuning and fine regulation that makes the design process even more complicated. But there's a new synthesis company called Avery that's working out of the Venture Institute right now that could increase writing the genetic code by 10,000 times. So that changes this arduous process of making a synthetic chromosome and testing it as a one-at-a-time event where we can make thousands of versions with variants and then screen for the life form that we want. So I think that's going to speed this research up maybe tenfold or more. So I think we can do it faster than it was done the first time.
Just before we take another question, Tom, are there any questions online? I'm not good with this technology. Is there a way of knowing whether?
Yeah, there is one hand raised up there.
Okay, and it's just that we are beaming this to the Stem Cell Society in Germany as well as Japan, and we want to give our partners. And there's Dr. Heschler, who is on our selection committee, so he helps select your prize. Dr. Heschler, it's good to see you.
Thanks so much that I have a chance. So very great lecture, Craig. I really like that very much. So my question is, you mainly talked about the genome. Can you understand me? Yeah, my question is, what is the state of the art on the intron? So you mostly talked about exons, the genes, but we know it's maybe only 1 or 2% of the whole gene sequence. Do you also have more information about introns and the the function of introns for life and for cellular
The
functions?
mycoplasma genes don't have any introns. When we look at the
Mhm.
first
Mhm.
archaea, they had relatively small introns, and so that's
Mhm.
certainly one step. But
Oh,
just
okay.
to have introns, then we have to have in all the splicing mechanisms and all those process spliceosomes. So that's where it becomes, you have to know all the components involved if we're going to add them back. And that's where we're surprised just with cell division. What people thought were the key
Mhm.
genes were only 2 out of the 7 that were. So it's definitely one of the next steps, because if we're going to get to a eukaryotic cell or even an archaea,
Mm.
we need to have those mechanisms.
Okay, thank you. Thank you so much.
Okay, I think because the time is late and I hate to cut this off, but you can approach Dr. Fender, and then I know the students will be spending an hour with him. Thank you again and congratulations on your tremendous science journey. Thank you.